Welcome back, everyone. Today we are taking a deep dive into a fascinating topic. We've got some excerpts from two scientific papers to guide our discussion. One compares the Aleppo pine to the Brucia pine and their hybrids in terms of drought response. And the second paper delves into the molecular mechanisms of drought, stress, and recovery in the Aleppo pine. But, okay, let's start with the basics.
Okay.
The first paper really sets the scene by talking about the drought challenges in the Mediterranean. And it emphasizes how the Aleppo pine is way more successful in these dry conditions compared to the Brucia pine. But the paper explains that the Brucia pine is more of a water spender, if you will.
I see. So it takes in and releases water pretty readily, which works fine when there's plenty of water around.
Exactly. But when a drought hits, it becomes a liability.
So what about the Aleppo pine? How does it handle things differently?
Well, it's more of a water saver. It closes its stomata early on to minimize water loss. Kind of a conservative strategy. The paper also touches upon the fact that these two pine species naturally hybridize. So that's where things get even more interesting. Some of these hybrids actually exhibit something called hybrid vigor. But okay, let's move on to the second paper and this plant array system. So imagine this. You have a bunch of potted plants, each sitting on a super sensitive scale. And because the weight changes are mainly due to water loss through transpiration, it's like tracking a tree's sweat rate, so to speak.
So we can actually see how much water each plant is losing in real time.
And it gets even better. From these weight measurements, researchers can also calculate stomatal conductance, which tells us how open or closed the stomata are.
Oh, so we can actually see how the plants are regulating their water loss at the leaf level.
Precisely. And they also determine a measure called the physiological critical stress point. It's denoted by the Greek letter theta.
Theta, what does that represent?
It's the threshold of soil water content at which the plant starts to experience significant stress and closes its stomata to conserve water.
So basically, it's like the tree's internal alarm bell going off. Time to switch to survival mode.
Exactly. And what they found is that the Aleppo pine has a significantly lower theta compared to the Brucia pine.
Meaning it can handle much drier conditions before it starts freaking out. Fascinating. So this plant array system, it really gives us a deep dive into how these trees are coping, huh?
Absolutely. But hold on, there's more. This second paper doesn't just stop at the plant array data. They also delve into the molecular changes happening within the Aleppo pine during drought and recovery.
Oh, so we're going even deeper, down to the genes themselves.
Yeah, they use a technique called transcriptome analysis. It's basically like taking a snapshot of all the genes being expressed in a cell at a specific moment. Think of it as a genetic activity log.
Okay, that makes sense. So they can see which genes are being turned up or down in response to the drought.
Exactly. And they found some pretty remarkable things. For one thing, they found thousands of genes with altered expression levels. But perhaps the most intriguing discovery was the activation of, get this, jumping genes. They're mobile genetic elements that can actually move around within a genome, like little genetic nomads.
Whoa, that's wild. So they're not just static, they can actually change position.
Yeah, and by doing so, they can potentially influence the expression of other genes. But I guess the big question is, how do we connect this jumping gene, this TNT194, to the actual drought resistance we see in the Aleppo pine?
Okay, so think of it this way. These jumping genes, they can act like genetic switches, flipping genes on or off, maybe even fine-tuning their activity, just by inserting themselves into different spots in the genome.
Okay, so in the Aleppo pine, maybe this TNT194 is messing with genes that are directly related to drought tolerance.
Exactly. It could be boosting the expression of genes that help the tree conserve water or protect itself from damage when things get dry. Or, on the flip side, it might be suppressing genes that make the tree more vulnerable to drought. It's almost like the tree is rewiring itself at a genetic level to survive. Pretty amazing, right?
It is. And you mentioned earlier how the researchers found thousands of genes with altered expression levels in the Aleppo pine during drought. Anything else jump out at you from that data?
Well, it's not surprising, but a lot of the genes that were affected had to do with photosynthesis and growth, basic functions.
Makes sense. If you're struggling to survive a drought, you probably don't want to waste energy on growing.
Right. It's like the tree is putting those functions on hold, conserving energy to focus on survival. And then when the drought ends and things start to recover, you see those genes involved in repair and growth getting ramped up again.
So it's a dynamic response, constantly adjusting to the environment. And this is where the plant array data comes in, right? We can actually see these molecular changes reflected in the tree's behavior.
Absolutely. The two data sets, they complement each other perfectly. Like, for example, we see a decrease in transpiration rates during drought. That makes sense, right? But then when we look at the transcriptome data, we see that it coincides with a downregulation of those photosynthesis genes.
Oh, wow. So we're literally connecting the dots between the genetic activity and what's happening in the whole tree.
Yeah. And it's the same story during recovery. The tree starts rehydrating, those growth genes get turned back on, and we see a corresponding increase in transpiration and stomatal conductance thanks to the plant array.
It's a beautiful illustration of how all these different levels of analysis can paint a more complete picture. But speaking of the plant array, we haven't really talked about how they actually measured that physiological critical stress point you mentioned earlier.
Well, remember how the plant array is constantly tracking the weight of each plant? That gives us a real-time measurement of transpiration, right? So as the soil dries out, the plant eventually hits a point where it just can't keep up. The transpiration rate plummets. And that inflection point, that's the theta.
So it's like finding the breaking point.
Yeah.
The moment the tree realizes things are getting serious and it needs to really clamp down on water loss.
Exactly. And what's cool is that the plant array is so precise, it can pinpoint that moment with incredible accuracy, giving us a really valuable tool for quantifying drought tolerance. And remember how we were talking about the Aleppo pine having a lower theta than the Brucia pine?
Yeah, meaning it can tolerate drier conditions before it hits that critical stress point.
Right. And the hybrids, remember them? They showed a theta similar to the Aleppo pine.
So potentially, those hybrids could be a really valuable resource for forestry and conservation efforts, especially as drought becomes a bigger issue in many parts of the world.
Absolutely. But, like we've been saying, theta is only one piece of the puzzle.
Right, we need to consider all the other factors too. Growth rate, disease resistance, the whole shebang. But focusing on the theta for a sec, is it a really complicated calculation to figure out from the plant array data?
Actually, it's surprisingly straightforward. It's all about finding that point where the transpiration rate takes a nosedive. The plant array makes